My name is Nathaniel Hawkins. I'm with the Department of Computational Mathematics, Science, and Engineering at Michigan State University, and today I will be presenting on our work titled Systematic Tissue Annotations of Genomic Samples by Modeling Unstructured Metadata. Currently, there are approximately 2 million genomic samples and other omics data from human and model organisms publicly available in repositories such as ArrayExpress. In the ideal situation, researchers would be able to curate samples and data sets that are relevant to some question of interest and reuse these data rather than investing resources into conducting additional experiments to collect new data. However, being publicly available as they are in these repositories does not necessarily mean that they are accessible. Data reuse is often impeded by the fact that metadata associated with these samples, these data, are described using natural language text in unstructured fields, which makes retrieving relevant data a challenge. Additionally, a vast majority of omics samples are missing basic information about sex and age of the individual they're collected from, the tissue of origin, disease states, treatments, etc. There are existing methods for annotating biomedical samples, and they sort of broadly fall into two categories. There are omic-based approaches and text-based approaches. Omic-based approaches utilize the underlying data to make some kind of metadata prediction. So, for example, this could look like taking gene expression data and using that as the features for training machine learning models to predict tissue annotations, for example. Methods of this type have been very successful for a variety of annotation tasks. However, they're limited by their data type specificity. So a model that's trained on something like gene expression or RNA-seq can't then be used to make predictions on high-C data. So for separate data types, models need to be retrained or perhaps even rethought. Text-based methods sort of circumvent this data type specificity limitation, and they apply principles from natural language processing to sample metadata in order to predict annotations. The benefit to doing this is that anything that has text can then be annotated using the same methodology, or a similar methodology at least. In our approach, we use sample descriptions from publicly available data in ArrayExpress, as well as a Python library called Flare to create numerical representations of sample descriptions and use those as the features for training logistic regression, one versus the rest classifiers in order to provide tissue annotations and cell type annotations for these samples. We're comparing our model predictions to two existing methods that represent classes of methods. There's Tagger, which is an NER tool, and MetaSRA, which is a prior knowledge and ontology-based tool. The comparison of the results for these for our work is shown in the middle of the poster here. Above that is a workflow diagram that I would be happy to talk about in more detail with anyone that is interested in some of the specifics of our pipeline and our workflow, including things like our text pre-processing, how we're weighting words in a sample description in order to provide perhaps more biologically relevant sample descriptions, and some of the more nuanced details of the training of the model, such as the cross-validation scheme to ensure that experiments are never split across folds. As I said, Tagger is sort of a named entity recognition tool, and it's looking for direct matches in the text to some kind of dictionary of ontology terms. The bottommost box on this box plot, represents the average precision of each of the models, and the model here being each of the Tagger terms that we are searching for. We can see the average is relatively low. Above that, we see the results from MetaSRA. I should clarify here now that each point in the box plot is a single model or single tissue that we're trying to provide annotations for. We see MetaSRA outperforms Tagger. As I said, MetaSRA is a tool that uses some prior knowledge, and it actually does a process of constructing a knowledge graph. Our natural language processing based machine learning models, which are just logistic regression classifiers based on word embeddings, outperform that. However, they are unable to beat the performance of models trained on the gene expression data itself. This is okay in our opinion because the natural language processing based models are much more widely usable to other data types, which is a limitation that we've already pointed out. The interesting result as well is that the top two box plots show combination of model results, whereby we take the predicted probability from the gene expression logistic regression classifiers, the natural language processing based machine learning classifiers, and MetaSRA, and we weight those by the F1 score in order to combine the predictions to an aggregate probability. And we see that adding the text-based methods to the gene expression predictions actually improves the performance further, to where if we combine MetaSRA, NLP, and gene expression together, the performance is above 0.9, which is showing that the text-based methods capture features that aren't necessarily seen in the gene expression data alone. Unpacking these models a little bit, we've done some work in interpretability of these models. On the right, we have a TSNE plot of the model coefficients. These are normalized, and they're colored as well as indicated with different markers by their high-level anatomical system. We see these models actually cluster into meaningful biological groups, which is an interesting result. We've also taken additional text from biomedical ontologies and turned those into word embeddings, similar to what we do with the sample descriptions, and made predictions on those. Here we're showing some of the top predictions for the brain model on disease ontology terms, and we can see that these terms match up with sort of expected brain identified diseases. One of the things that we're working towards in the future is creating a web server whereby we will host all of our predictions for publicly available data so that individuals can go and search and retrieve relevant data, as we indicated in the beginning as being sort of the ideal case for exploring novel biological phenomena and taking advantage of these publicly available data. Some initial screenshots for this web server are shown on the left here. We have text completion for the searches, which will match to available models using Elasticsearch. Thereby, individuals can sort of select the most appropriate tissue from the list of available models that we've made predictions for. Those query results are then listed by their predicted probability from our models, and individual samples and series can sort of be selected and downloaded later. If anyone is interested in talking about this work further, my contact information is shown here. Thank you very much.